All right, let's continue our discussion of chapters 12 and 13. I believe we left off here um, with the making of RNA, what's known as transcription. Um, you can think of this as essentially we are transcribing the code from, or rewriting the code from DNA to RNA. We're going to use DNA as a template to make a piece of RNA. And as you know, the DNA is contained in the chromosomes. Chromosomes are very long and have a lot of DNA in them. But when we transcribe, we're just going to transcribe a particular gene. So on that whole chromosome, we'll only transcribe a particular gene to make a particular piece of RNA that's based on that gene. You never transcribe a whole chromosomes. You just transcribe pieces of them, individual genes. Okay. Now, the main player here is RNA polymerase, the enzyme that carries out transcription. It essentially reads the gene, that reads the nucleotides in that gene, and builds up the corresponding piece of RNA that matches up with the bases in DNA. Note again, our RNA is single-stranded, and we have uracil instead of thymine. So essentially, as you build up the RNA where you would have put a T if you were building DNA, because we're building RNA, we put a U instead. All right, how does the RNA polymerase know where to go on that chromosome to transcribe? Well, there will be certain chemical signals that essentially are telling the cell we need to transcribe this gene. And in particular, there are regions called promoters, which are next to the gene that we're going to transcribe. And so the RNA polymerase knows to essentially attach to the promoter and begin transcribing that gene, or what you can see here, a fancy way of thinking of that, an RNA coding region. All right, so we're going to read the DNA again, which is double-stranded. We're only going to read one side of the DNA, what's known as the template strand. The other side will be ignored. And we make our piece of RNA, our RNA transcript. Okay. Now, in eukaryotes, as opposed to bacteria, so in us, eukaryotes, there's a step that you don't see in bacteria, and that's called RNA editing. What happens is when we make that initial transcript, we transcribe the whole gene, but in eukaryotes, that gene contains these things called introns. And these are parts of the gene that aren't really needed to make our final piece of RNA. And so we have to edit it by essentially cutting out these introns. What you have left are known as the exons. The introns get removed, the exons are spliced together, and you have your final piece of RNA, in this case an mRNA. Processing or RNA editing also entails putting on what's called a cap or a tail, and the cap and tail essentially protect the mRNA transcript, and when it comes time for translation, they tell the ribosomes where to start. They'll always start at the cap end of the mRNA. All right, so let's think about what we're going to do with this mRNA now, and we're going to talk about translation. Remember, with transcription, you're using DNA to make RNA. You're essentially rewriting the code. But think of it this way, when you go from DNA to RNA, you're in the same language. You're just transcribing it, rewriting it. But with translation, what we're going to do is we're going to use RNA as a template to make a protein. Okay? We're not converting RNA into protein. We're, again, using it as a template. But now we're translating it, we're changing it from the language of nucleic acid to the language of a protein or a polypeptide. All right, so how does that happen? Well, in our RNA, in particular our mRNA, we have these things called codons, and codons are batches of 3 nucleotides in the mRNA that are next to each other. So here's one codon, here's the next codon, etc. And these codons essentially can embody the genetic code. The codons tell us which amino acid is going to go in this position on our protein, which amino acid is going to go here, and which amino acid is going to go here. So essentially a codon codes for an amino acid. And here is the genetic code. All right. When you have a series of codons, like we have up here, we have one codon that's AUG, one that's AAC, and one that's UCU. We look in the genetic code to figure out, okay, which amino acid does AUG code for? And so in your codon, you have 3 nucleotides, the first one, the second one, and the third one. And so we look on the side here, okay, we have AUG. So our first codon is A, our second codon is U, and our third, notice now, this gets us into this box right here. Now we look over here for our third codon, AUG, and that corresponds with methionine. So essentially our first amino acid is methionine. We'll abbreviate that MET. Our second one is AAC. So let's look what that's going to be. So now we're at AAC. So we're right here, and it's ASN is our next one, which I believe corresponds with an amino acid called, no, this is aspartic acid, so ASN, it's arginine, ASN is, I can't remember what that one is, we'll look it up. All right, our next one is UCU, so let's look up that one. All right, what did I say, UCU? So UCU would be here, here, and here, UCU, so that gets us to the amino acid serine, S-E-R. So this stretch of mRNA codes for the amino acid sequence MET, ASN, serine. Okay? So that's how you use the genetic code. We'll practice that more in class so that we know how to use it. Excuse me. All right. But now how exactly does translation happen? All right, well, first of all, we need 3 types of RNA. We need the mRNA, which contains our codons. We need the rRNA, which is going to comprise our ribosomes. And we need the tRNAs, which are going to carry amino acids to the ribosome. All of those are made through transcription. So the mRNA, well, all the RNAs are made in the nucleus. They travel out of the nucleus. The ribosomes, again, are comprised primarily of rRNA, but also some proteins. The mRNA gets together with the ribosome, and the ribosome essentially reads the codons on the mRNA. The tRNAs bring the amino acids, and notice there's a group of 3 nucleotides on our tRNA that matches with the codon. So AUG, again, we know, corresponds with methionine. And there's a particular tRNA that brings methionine because this tRNA has the what's called the anticodon that matches up with the codon, AUG. And so the anticodon that matches up with AUG is UAC because those are the appropriate corresponding bases. The next tRNA comes along that matches up with the codon UUC, and it's bringing phenylalanine, as you can see. The next one, the codon is AAA, and the anticodon on that matches up is UUU, and that tRNA brings lysine. Now notice they differentiate what's called the start codon here. The start codon is the one that always signals the beginning of an mRNA, and it always corresponds with AUG and methionine. So that's what always begins the production of the polypeptide by the ribosomes. So the mRNA just moves along the ribosome. The appropriate tRNAs bring on the appropriate amino acids. The amino acids are bonded together with peptide bonds, and the protein grows, or our polypeptide grows, until finally you get towards the end of the mRNA and you have a stop codon. And notice up in our code there are 3 different stop codons, AUU, AUG, and UGA. So where you get to that on your mRNA, that signals to stop, and the ribosome knows to stop translating, and our protein is finished, and it will break off and perhaps be processed in the Golgi apparatus and then do its thing. All right, so let's review this here now. The mRNA, again, all these RNAs are made by transcription in the nucleus, but the mRNA basically has the codons that determine the amino acid sequence in our polypeptide. Our RNAs comprise the bulk of the ribosomes, which are the site of translation, where we're going to make the protein. tRNAs bring the amino acids to the ribosome, and remember, tRNAs contain the anticodon that matches up with the codon on the mRNA. So all of this comprises a very important process in living things and is, as we say here, the basis of heredity. That is, we've got DNA that has all these nucleotides, these genes. We use the DNA as a template to make RNA. The RNAs of various kinds are then used to make proteins, and the proteins are basically what do all the work inside of our cells, and they also comprise a considerable amount of our structure. Think muscles. Muscles are made of proteins. And again, to make those proteins, any kind of protein, you have to make use of RNA, which is made using, again, DNA as a template. So this, you could also say the proteins is sort of the expression of our hereditary because they do all that work. We'll talk later about how when you have mutations in the DNA, that can lead to proteins that are defective and can lead to problems, and that's what we see in a lot of diseases that are inherited. Is due to inheriting mutated genes that leads to a defective protein, which causes some kind of problem. All right, we'll stop there and do the rest of it in another video.